You know, there's this hidden layer of chemistry that connects all sorts of things you'd never think were related. We're talking about our blood, the green leaves on trees, even powerful, life-saving medicines. And it all comes down to the secret life of metals and a really special class of molecules called coordination compounds.
So let's start with a bit of a riddle. What could a cancer drug, the red stuff in your blood, and the green in plants possibly have in common? I mean, you've got a heavy-duty chemo drug, the molecule that's literally carrying oxygen to every single cell in your body right now, and the engine that powers photosynthesis for basically all plant life on Earth. What's the link?
Well, the answer is that they are all a special kind of molecule called a coordination compound. At the very heart of each one is a metal atom, platinum, iron, or magnesium, and it's holding on to a specific group of other atoms. And it's this unique structure, this precise arrangement, that is the secret to all their incredible powers.
But for the longest time, these compounds were a total puzzle. Chemists knew they existed, but their structures just didn't make any sense based on the rules of the day. To really understand how we cracked this chemical code, we have to jump back to the late 1800s and meet a Swiss chemist named Alfred Werner. He was the one who first started to make sense of it all.
So, the first big clue came from just seeing how these things behave in water. If you take a simple double salt and dissolve it, it just breaks apart completely into a soup of simple ions. It totally loses its identity. But these coordination compounds, they were different. When you dissolve them, their core structure, that metal with its attached atoms, it stayed perfectly intact. It was like a little team that refused to break up. And Werner gave this stubborn little core unit a name, the coordination sphere. I want you to think of the central metal atom like a VIP. And the molecules or ions that are directly attached to it, we call those ligands, they're like the VIP's personal entourage. This whole group, everything inside those square brackets, moves as a single unit. Anything hanging out outside the brackets, that's just a counter-ion. It can come and go.
Now, to explain this, Werner came up with an absolutely brilliant idea. He proposed that the metal actually has two different kinds of bonding power, or valency. The first one, the primary valency, is just the metal's charge, its oxidation state. That's pretty standard, and it gets balanced out by those counter-ions. But the second one, the secondary valency, was the real game changer. This is the number of ligands in its entourage, and it's non-ionizable. It's fixed, it's unbreakable, and it's what dictates the whole complex's 3D shape. This idea was totally revolutionary.
Okay, so Werner's theory was great for figuring out the basic structure, but it didn't explain everything. It left some big questions on the table. Like, why do these things have such specific shapes? Why are some of them magnetic? And the biggest mystery of all, why do they have such beautiful, vibrant colors? For that, we need to look at two theories that came along later.
So, two major theories stepped in to try and fill these gaps: valence bond theory, or VBT, and crystal field theory, or CFT. VBT is really good at predicting the 3D shape of the molecule. It looks at how the metal's orbitals kind of mash together to form bonds. But it has this one massive blind spot. It has absolutely no way to explain color. Then you have CFT. It takes a totally different route. It basically ignores the covalent bonds and just focuses on the purely electrical interaction between the metal and the ligands. And that simple, elegant shift in thinking, that unlocked the secret to their color.
A really simple way to think about it is this. VBT gives us the skeleton, the physical shape of the molecule. But CFT gives us its soul, its vibrant color.
All right, so the absolute heart of crystal field theory is this one concept called d-orbital splitting. You see, if you have a metal ion just floating by itself, all 5 of its special d orbitals have the exact same energy level. They're what we call degenerate. But the moment those ligands start to approach it, everything changes. Let's just walk through this. Picture 6 ligands closing in on our metal ion. The electrons in those ligands create an electric field, and this field repels the electrons in the metal's d orbitals. But here's the trick. The repulsion isn't equal. The 2 orbitals that point directly at the incoming ligands, we call them the EG set, they feel this repulsion the most, so they get pushed up to a much higher energy level. And the other 3 orbitals, the ones that are pointing between the ligands, the T2G set, they're less affected, so they actually drop to a lower energy level. And that energy gap that just opened up, that is the crystal field split. Now, this whole energy split thing might sound super abstract and theoretical, right? But I promise you, it is the key. It is the fundamental concept that unlocks everything, from the deep color of a ruby to the way a life-saving drug actually functions. This is where the theory hits reality, hard.
Chemists have a symbol for this energy gap. They call it delta O, And the precise size of this gap, how much energy it takes for one little electron to make the jump from the bottom level to the top level, that is what determines the exact color of the compound. What's so cool is that different ligands, different entourages, create different sized gaps. And that's why different coordination compounds have all these different amazing colors.
So here's the play-by-play. White light, which contains all the colors, shines on the complex. The compound then absorbs just one specific color of light, the one whose energy perfectly matches that delta O gap. An electron grabs that little packet of energy and uses it to jump up to the higher orbitals. All the rest of the light just passes straight through. And what our eyes see is that leftover light, which just happens to be the complementary color to the one that was absorbed.
Let's take a classic textbook example, this titanium aqua complex. It's got one d electron just sitting there. It turns out that it absorbs light right in the yellow-green part of the spectrum. That's the perfect amount of energy for its electron to make that jump. So if it's taking out the yellow-green light, what's left for us to see? The complement, a beautiful, deep violet. And that is exactly the color of this complex. It's that simple and that elegant.
And that brings us right back to where we started. From cancer treatments with cisplatin to oxygen getting carried around by hemoglobin, and even in huge industrial processes like purifying nickel or something as common as testing your tap water for hardness. The unique, stable, and predictable structures of these coordination compounds make them absolutely essential tools across all of science and nature.
So this all leaves us with a pretty powerful thought, doesn't it? If we can understand these fundamental principles so well that we can literally design custom molecules to go in and target cancer cells, what comes next? What other massive problems could we start to solve by using the incredible, versatile rules of coordination chemistry?